Rohit sir, are you there?
Yes, ma'am.
Sir, am I audible properly?
Yes, ma'am. Clear. Crystal.
Earlier I was, earlier I was audible or I was not clear.
No, it's clear. Crystal clear, ma'am. Audible.
Okay, thank you, thank you.
Dr. Jyoti ma'am,
Yes, sir.
you are kindly requested to please proceed opening remark. It's 6:30.
Should I go ahead?
Firstly, Dr. Jyoti will proceed introductory remark. Dr.
Okay.
Jyoti, are you there?
Yes, sir.
Then kindly proceed, madam.
Should I?
I'm reminding you again and again. Kindly proceed.
Okay, sir. A very good evening to one and all. Today, I, Dr. Jyoti Sharma, Organizing Director, welcome you all in 30 days international training from Winter School on navigating agriculture technology transformation for upliftment of rural excellence, organized by AgriMeet Foundation Bharat in collaboration with Southern Federal University Russia, CIMMYT Mexico, ICREDA Lebanon, ICRISED Hyderabad, Manage Hyderabad, Shrikonda Lakshman Telangana, Horticulture University Telangana, ICR IRI New Delhi, MHU Karnal, IGKV Raipur, MGUVV Durg Chhattisgarh, ICR DCR Puttur Karnataka, ICR Atari Jabalpur, Stanford University and University of Oxford. These are the collaborators we have for our trainings. So with this, I would like to welcome our today's speaker, Damaris Odani ma'am. She is a well-known plant molecular breeder, currently a global research cluster called Genomics, Rebreeding, and Bioinformatics within the Accelerated Crop Improvement Program at International Crop Research Institute for Semi-Arid Tropics. She had led and implemented genomics project in both cereal and legumes, including the development of genomic resources in several underutilized crops. Damaris ma'am worked closely with other disciplines from national, private, and international institutes to develop the cutting-edge genomic resources for all ICRISAT mandate crop, that is sorghum, pearl millet, finger millet, groundnut, chickpea, and pigeon pea, in her current role. Prior to this, she worked as a senior researcher at Agriculture Research Council, South Africa, where she implemented molecular breeding for several indigenous crops like sorghum, cassava, but sweet potato, and many more. Damaris is also very passionate about mentoring of young upcoming scientists and has successfully mentored more than 50 scientists in her career up to date. She leads the Science and Publication Subcommittee of African Plant Breeding Association. Uh, Lot more to say about ma'am, but with this, I like to welcome ma'am for the today's session. And her lecture is on trade discovery in dryland crops. Ma'am, we would be very pleased to invite you on the behalf of AgriMeet for the today's lecture, and our participants will be very happy to hear from you. As you are very expertise in a research from South America, you have worked in many countries. So we welcome you for today's session.
Thank you. Thank you so very much for such a wonderful welcome. Thank you also for the opportunity to share the little knowledge that I have with everybody. I will get right into the presentation. Since you've done such a wonderful introduction, I don't need to say any more. I'm only going to make one request. I'm sitting in a very remote location in Tanzania. The internet is not very good. So I'm going to turn off the video and I will turn it on again during Q&A session if there'll be any questions. So just to enhance the experience of the presentation, I'll turn it off and then you'll see me again towards the end. I would want to say that it should be as interactive as possible. So if there's any question, I'm quite happy if you interrupt me. and seek clarification. But somewhere in the middle, again, I'll stop and ask if there's any question. But I'll intend to do about 45 minutes of the lecture, and then we can have a few minutes for Q&A. Now, going right into it, just for those who may not know about ICRISAT, you can go to our website, icrisat.org. We work across semi-arid tropics. We work on dryland crops. We work on resilient crops. Also, very nutritious crops. We work on those 6 major crops, but we also work on several minor millets. Our presence is across the globe. As you can see, the varieties that we have released across different continents, different countries. And so if you want to know more about ICRISAT, you can go to icrisat.org for more. And thank you for allowing me to choose the topic. And this is one of the things that I really I'm passionate about talking on dryland crops, because we know that currently there's the threat of climate change, and we are experiencing rising temperatures, especially in Asia and Africa. And this picture here is showing how, if you look from 1963 to about 1982, you see the temperature is more or less stable. It was moving up and down, but it was more or less stable. And then it started to shoot. And now we are on an upward trend, which is very, very worrying. And so for that reason, we need to build the required systems in place to ensure that we are able to deal with this kind of rise in temperature. And if we do not, we are likely to face a really terrible crisis in the future. And the way that you define dryland crops, these are the crops that are able to thrive despite extreme climatic conditions. And some of the things that are expected to happen because of the rise in temperatures, we expect more heat periods, much longer heat periods. We expect more droughts, and we're already experiencing them. More salinity tolerance will be required, because a lot of flooding would end up causing salinity in many cases. We also expect to see a lot of pests and diseases, and we're already experiencing them. Things like fall armyworm, which has suddenly become such a big problem. We expect that will happen with rises in temperatures across these semi-arid regions. So the crops that I'm going to talk about, or the strategies that I'm going to talk about, are very specific to dryland crops, but essentially can be used across several different crops. Now, we have a very, well, not that we do, the general trait discovery pipeline remains the same, irrespective of whether it's dryland crops or other crops. And the picture I'm giving there is the strategy that ICRISAT is applying, especially because our work is in collaboration with national partners, and we are our biggest consumer is the smallholder farmer. So the regions where we work in the semi-arid tropics, most of the farmers are smallholders and they're subsistence farmers. And so our work is basically to boost their production and to enhance their experience. In most cases, get them out of poverty and get them into food security. So the pipeline that we use in general starts from characterizing germplasm, and then using that to discover resilience traits. And I've already talked about what these resilience traits are. Once we discover that most of the resilience traits are quantitative traits, but we do look at how to introgress them into farmer preferred backgrounds once we identify and discover them, and then develop varieties from them. And the way that we work is together with national partners from different organizations like ICAR, IIMR, yeah, so different national programs, but also with funding organizations such as DBT, DST, Bill and Melinda Gates Foundation. We work together also with several different disciplines, and we integrate capacity building in the way that we do our discovery. Now, since this is a lecture, I'm going to just take the trait discovery component and the two approaches that we use, and then just give a few strategies that we use with respect to trait discovery. And this slide is just showing some of the prerequisites that you need to be able to carry out a trait discovery experiment or a project, trait discovery project. Now, traditionally, like let's say 2 decades ago, we could have done this not very easily. We could have still done it without a reference genome, but because generating reference genomes has become so affordable, it has become one of those things that if we want to do trait discovery, it is essential, because we've moved from doing a few SSR markers into doing genome-wide markers. And so for you to do a trait discovery in dryland crops, for resilience trait, we typically use, so we start with a reference genome if we do have, and if we don't have, we can use a related species, but we tend to screen gene bank material. And this is especially because when it comes to resilience traits, gene bank material or wild relatives or land races tend to have more resilience than the cultivated. So it's always a go-to resource. We can also use breeding lines, and then after we identify what we are going to work with, then we screen for the trait of interest. And we start with a larger pool of germplasm, we screen for the trait of interest, and then we can decide to go ahead and validate that trait in a much smaller biparental or a few multi-parent populations, and then do genomic characterization of the regions that are associated with the trait, and then go into using it for developing varieties. So this is a very broad outline of how we do trait discovery in dryland crops for resilient traits. Now, the typical types of populations we use are either a diverse set, as I mentioned, we can go to the gene bank, or if we've already identified some very good genotype that is excellent in that particular trait that we are trying to discover, we can develop a biparental population between the genotype that has the trait and the one that doesn't have the trait, or maybe lower in expressing the trait. And so you create different numbers of recombinations that will help you to associate some segregation types with certain regions of the genome. We could also use multiparental populations. So for example, if you're looking for tasty, drought-tolerant sorghum that is red and resistant to birds, so then you might end up with one genotype that is really the right color, another one that is really drought-tolerant, another one that is resistant to birds. I see someone has raised their hands. Please go ahead. Did someone want to say something? Okay, I'll proceed. So you then use these several different genotypes that have unique traits and create multi-parent populations. Now you'll end up with a diverse set of populations having unique backgrounds, but that are segregating for all the traits that you're looking for, and then you can end up studying that trait of interest. So we typically use these 3 major different types of populations. If you use a diverse set or multiparental populations, you end up doing what we call genome-wide association studies. It has become a very common word in trait discovery. For the biparental, you typically do biparental QTL mapping. And I'm going to go through what it entails, how you select the parents, everything. I'm going to go through that in the next in the next steps. So once you've figured out your populations, now you go into beginning setting up your experiment. So let's say we decide we are going to use a diverse set of population. You've identified them, you've obtained the material either from the gene bank or you've obtained it from different breeders. So if you're looking to do a genome-wide association study, which is basically looking at different unique recombinations across this diverse panel that would be associated with the trait, then you end up doing what is called a GWAS. And if it's something that you've identified across two different parents, one that is really, really good in that particular trait and another one that is low in that trait, you create the biparental or multiparental populations. Now, I'll start by just defining what GWAS is. So this is genome-wide association studies defines looking at associations of molecular markers, simply, certain variants in the genome. And in this case, we're talking about SNP, single nucleotide polymorphism markers, because they have become very robust, genome-wide, and very simple to generate, and very simple to score as well. So genome-wide association studies basically defines the association of those specific molecular markers with a trait of interest. And usually this would have been scored across a wide collection of individuals, as I just mentioned. A pictorial view is just, looking at, and the example I'm giving on this slide is for finger millet. So for example, you probably have 1,000 unrelated finger millet individuals that is maintained somewhere. And what you do is you phenotype them across different environments and you score for blast disease. In this example, I'm giving blast disease as the example. So you phenotype them across different environments and across different locations or different seasons, and then at the same time genotype or generate genome-wide SNP markers for those same 1,000 unrelated individuals, and then look at the association between the markers, molecular markers, and the phenotypes that you have generated, and then co-associate the markers and the phenotype. So that basically defines what GWAS is. Now, I've talked about using diverse parents versus using, family-based mapping, where you have 2 parents or a few parents. What is the difference? What are the advantages? Now, association mapping is you're taking advantage of the natural recombinations that exist out there in nature. And so it's something that you probably don't have to initially invest much on. You just go to a diverse set that's already existing, take them, and look at natural variation for that trait of interest. For that reason, you can do it for trees, which usually takes a long time to develop mapping populations. You can do it for crops that are impossible to cross. You can do it for also clonally propagated plants. You would imagine it will account for more phenotypic diversity because you're tapping into what exists in nature. And that enables you to be able to explore a lot more recombination events. The only challenge with that is that you don't know how far back that recombination would have happened. And in most cases, we probably don't even need to know. But in some cases, it might be useful to know if you want to know how long this trait has existed. When did the mutation occur, if it's a mutation that happened? But also in some cases, and in very many cases, you might end up with false signals. And I don't know if it's just me, but we've ended up with quite a number of genome-wide association studies that have been published over the last several years. And sometimes you look at them and you're like, there's something that is not right about the associations published here. So sometimes it's either too stringent and then you end up missing the association that you're looking for, or you use a model that does not fit for the trait that you're looking for and you end up with just false signals. So that's something that is important to think about if you're talking of genome-wide association studies. But if you compare using a diverse set of populations and pedigree-based mapping, pedigree-based mapping gives you a better resolution, but it takes relatively longer to set up the population. So, you have the option of compromising maybe the resolution and using the immediate diverse set that you have, or investing a little bit more on generating the mapping populations and improving the resolution of your mapping. It really depends on the resources you have and the time that you have also, and how accurate you want it to be for your breeding program. Now, the prerequisites for association mapping is, there has to be linkage disequilibrium. And so it means that there'll have to be some kind of association between the trait you're looking at and the markers that are present within the population that you're looking at. You also need to establish the relatedness. Even though you're working with a diverse set, we do know that there would be some relatedness across these different genotypes. So look at how they are clustering. Are they clustering according to where they was collected? Could that have some influence on the expression of the trait you're looking at? And then take care of whatever relatedness is there, because you don't want to have false signals that is just merely based on the fact that these few genotypes have come from the same ancestor. So you need to take into account things like kinship metrics or principal component analysis that you use to correct for relatedness. But you also do need to have robust numbers of markers. As I mentioned, we've moved from a few markers across the genome. 20 years ago, you could get away with doing an association mapping with 200 markers. Right now, it probably won't work really well because we do have quite a bit of resources in terms of molecular markers that you can use to enhance the resolution or the association that you're looking at between your markers and the phenotype. But one of the things that is usually underrated so much in genome-wide association studies is the phenotyping. Phenotyping. And this is so crucial that you do need to have reproducible and reliable phenotypic data. If you do not have reliable, reproducible data, you are actually wasting time in genotyping that data set in that population in the first place. And I'll just give an example. We do a lot of GWAS studies, and sometimes we receive data sets that someone is asking you, I've phenotyped this population. I do know you have generated markers for them. Would you mind doing for me a GWAS analysis to see if there's any set of markers that are associated with the trait? And then you sit down with this data set and you look and you see there is heritability of less than 10%. And that means that the phenotypic data set that is provided is not reproducible. Or sometimes you just have such high differences in locations or in environments, they are not consistent. When you have such data, please do not waste resources generating genotypic data or even attempting to force GWAS to work. Because at the end of the day, the reason you're doing association mapping is to be able to identify something that eventually you can use to develop an improved variety for that particular trait. And so what is the point trying to force random alleles to get associated with a phenotype that is not consistently inherited or is not phenotyped properly, and so the data set is questionable. So just to stress that you need to have data set that is reliable and reproducible. And I do know that there are studies that accept even as low as 0.3, 30% heritability. We tend to be very strict with this and just have analysis done for highly heritable, at least populations where the phenotypic data set shows good heritability, at least 60%, and in most cases, that will give you a really, really, really good association. So I'm just giving an example here of diverse genotypes that you could use. This is an example on groundnut where we looked at different market types. We looked at Virginia, Spanish, Valencia. So you can see, we have a good enough representation of the different market classes. It's diverse. The markers that we're working with also, as you can see, on this, the principal component figure here, that the markers are very informative. And that's the other thing. I didn't mention it before. When you generate molecular markers, it's also important that the markers you're retaining for your association mapping, that they are informative. It doesn't matter if you have 10 million SNPs for that population. If they're not informative in differentiating the genotypes, you could as well throw them away. You could end up with just 3,000 markers that are super informative, and they will help you in terms of finding association between the markers themselves and the trait. And so one of the ways that you can detect that the marker set that you're using is informative is by looking in a principal component analysis like this one, the genetic variation explained by that set of markers. And as you can see in this particular study, the first, second, and third principal components are explained by more than 60%. The markers explain more than 60% of genetic variation across the individuals. That's a really good set of markers. But I know in most cases, people want to go with millions of SNPs and not even looking how informative are those SNPs. And you can even have 10 million SNPs, but if they're not informative in terms of differentiating your genotypes, it's just going to give you a lot of junk out of your association analysis. And so in this particular set, we can actually see there's some clustering according to market classes. And so we use this information to correct for population structure, because we can clearly see this population structure. Also, if you look at this the DAPC plot we have here, we can clearly see the clusters. So before you do the association analysis, you correct for those the structures so that there's no bias in the mapping based on the relatedness of the individuals. A good example of a data set that you probably don't want to use for association mapping is something like this. This is a set of genotypes that appear very, very, very related. As you can see, once you correct for the population structure here in this data set, you probably just end up with less than 100 unique genotypes. And so something like this, if you find, then your population is not representative enough. Most of it is quite relative. So for example, if you can see here, all these appear to be very, very similar. They are similar, actually. It's almost like the same thing. Here also, you see these are very similar. So this is an example of, you know, something that may not be good for association mapping, because there's just so much relatedness across the germplasm. And by the time you correct for the relatedness, you end up with almost nothing that is unique to work with. Now, I'm just going to give an example. You do need to have well-distributed genome-wide SNPs. Beyond the SNPs being informative, as I did mention, they need to be well-distributed across the genomes. And the examples I'm giving here are one where you have markers, you have several markers, 120,000. This is for the sorghum. I'm giving an example in sorghum. So you have about 120,000 SNPs, very nicely well distributed across the genome. It will do the work because you know every chromosome is covered. But I'm also giving another example here where you have fewer markers, you only have 8,000, but you can see they're still fairly well distributed across the genome. This also works. This works. And again, going back to what I was mentioning, you'd rather have 8,000 markers that are well distributed across the genome but are informative in differentiating your individuals than have 1 million of them that are probably skewed. So you might have just chromosome 1, 2, and 10 having most of the markers, and the rest are just scattered, and then the markers are also not informative. So, I mean, these are some of the things that you really need to take care of when you're looking at a marker set to use for genome-wide association study. Now, coming now to planning a study, I did mention looking at the population size. So what would be the ideal population size? Things like what is the experimental design? How are you going to do the phenotyping? And I'm going to give these examples based on dryland crops. As I mentioned, for most of the dryland crops, we're looking at climate resilience traits, and these traits are quantitative. And so I'll walk you through the kind of population sizes you need and the design and so forth. So the rule of the thumb is the larger the number, the higher the power and precision of mapping. And we also do know that we are limited in terms of how much you can phenotype. You can end up with 10,000 different accessions, but then phenotyping becomes a nightmare. So you have to strike a balance between the resources that you have and what you can manage with confidence. And in most of the traits that we're working with, a minimum of 200 is recommended. In fact, I'm just giving this example of a publication in Bali that was recommending at least 384, and that's in Bali. You could actually do a calculation for different species to be able to see the recommended size, which would be the bare minimum if you want to do a good association study. Yeah, and then the size also would depend on the trait to be examined. If you're working with a simple, if you're working with a simple trait, you can easily get away with maybe between 150 to 200 for sorghum. Sorghum has a relatively smaller genome size. But if you're working with a quantitative trait like drought, then the moment you're going below 200, it's already a little bit of a risk, because there's just so many things in the genome that are contributing to drought that if you end up with so few, you may not end up with a good association study. In some cases, even though you're working with a complex trait and you're just limited with resources, there are things that you can do to make that experience better. So you can start with a representative set and use that to see if you can detect association in a representative set that may not all have been phenotyped. I'm tending to see hands up randomly, and I would say if you have something to say, you can just go ahead. And when I hear you, I'll be quiet and give space for a question. But maybe I could stop here for a second and just ask, so far, are there any questions, any clarifications that anyone might need? Okay, I take the silence as no questions, so I'll proceed. In terms of experimental design, there are several experimental designs available out there when you're working with large population sets. The most important thing is that it should be replicated within the season, but also across the seasons as much as possible, across season and across locations as much as possible. And as I did mention, it can be one stage in which the same genotypes, you use them across the different seasons, different environments, or you can do two-stage, where you use a few individuals with the trait of interest and genotype them, and then only do another repeat association mapping with the markers that you found to be associated within the first stage. So in that case, I mean, this is particularly useful if you're saving money on genotyping. But for most part, genotyping has become the cheapest component. Phenotyping is the biggest challenge, especially when you're working with climate resilient traits, because they require quite specialized equipment, and also it's just very challenging to get precise data. Okay, so the next is, how do you do your phenotyping? And for you to get accurate associations for quantitative traits, it's recommended to avoid yes or no answers, because we are working with quantitative trait. It's not, as you look at this histogram here, it's not just yes or no. There's yes, maybe, yes, may not be, and so forth. So it's important to develop a scale that takes into account the continuum of the trait. And so, for example, you would hear recommendation for scoring diseases, for QTL mapping or association mapping, that it should be scored from 0 to 9, as opposed to from 0 to 5. And, there are certain cases where it might be too cumbersome, but if you want accurate association, make it a quantitative scale rather than 1 or 2, or 1 or 2 or 3, when you can take care of 1 to 9. And just giving an example here, this same trait that has a really nice histogram on your left, if you look on the right, if you just scored it as 0 or 1, 2, 3, 4, 5, look at what you end up with, and you end up not really taking care of the quantitative nature of that trait. So if you want to have a good association, go for a more quantitative scoring approach. In terms of genotyping, as I did mention, we've moved from a few markers, nobody's using SSRs anymore. It doesn't mean that you shouldn't use if that is all you have, but there are other options that give you better genome coverage, better and more even genome coverage. And we typically do scheme sequencing, because then you are having like reduced representation of the genome, but still ending up with really good coverage across the genome. In some crops, there are SNP chips available, so you can use the SNP chip. Something that is coming up now quite a lot is using targeted sequencing, and it's also pretty easy to use. And targeted sequencing is replacing the SNP chips that we used in the past. It is affordable, and every time you can generate really good data without necessarily having to having to do it all again every time, the way that you did it with the SNP chips. So it is quite affordable. If the only thing that you have to do is SSRs, why not? And when we're talking about climate resilient crops, we are also aware that in certain cases, the only thing that you might have is simple sequence repeats or AFLPs. Why not? I mean, if 20 years ago we were able to identify associations, nothing stops us from doing that as well now. So if that's the only option you have, yes, you can go for it. So in terms of data analysis, again, this depends on data that has been generated, but the two common methods are either general linear model or mixed linear model. Now, there's almost no study I see that's just using general linear model because it's too simplistic and does not account for relatedness. And for that reason, you end up with a lot of false associations, which you don't want. The mixed linear model is the best, is the most recommended because you're working with at least two markers that are looking at association with a particular trait. Now that allows you to be able to know the size of the region you're working with and to be able to eliminate false positives in a very cost-effective way. But in any case, since you do have both general linear models and mixed linear models, you can use both and present results that are consistent between general linear model and mixed linear models. So one of the very controversial aspects of genome-wide association studies has become how to define the threshold. And, traditionally, the p-value and Bonferroni was what was used. And, the Bonferroni was too stringent. And in most cases, people did association studies and did not end up with any associated or significant SNPs. And then we've now gone back into looking at different ways of looking at the threshold. But what I would say is it would be good to combine a number of things. And typically what we do in our group is look at the p-value and the Bonferroni, look at correlation between a pair of loci and the phenotypic variation explained, and also look at false discovery rate. So use at least two of those to narrow down on what you are confident is the association that you're looking at. And we choose the conservative approach because we don't want to carry on with things that are not confident. So we choose the conservative approach and use, for example, R-square value of at least 15.15. P-value, we still stick to Bonferroni. And so any of those two that gives us good results, we proceed with. But in most cases, we end up with really good set because also we will have taken care of so many things and only retain informative markers. And so we seem to do pretty well with Bonferroni and phenotypic variation explained. And we end up with really, really good haplotypes as well towards the end. Now, I'll switch and go into, because remember we're looking at trait discovery for climate resilient crops, both using diverse genotypes and biparental. So I've just looked at the diverse genotypes and I'm going to switch now and just give a quick run through how you do it using a biparental population. Now, the example I'm giving here is definitely the simplest, but you typically would cross genotype A and B. Both of those need to be contrasting for your trait of interest. Generate your F1s. It's important to confirm hybridity in your F1, and there are markers out there that you can use to confirm hybridity. And once you do that, you will self your F1 to end up with F2, and you can do your mapping at F2 if it's a simple trait, or you can advance your recombinant inbred lines up to at least F6 and do recombinant inbred line QTL mapping. So that's a very summarized way of explaining how you do the pedigree or family QTL mapping. There are various types that you can do backcross, you can do near isogenic lines. If you're interested, there are many publications out there. Please have a look. So I'll just carry on and give this example with the simplest model that has these two parents. Now, the type of population size will, as I mentioned for GWAS as well, the size of the population will depend on the trait. Now, because we have better resolution with the biparentals, it's sufficient to use between 150 to 200 as a rule of thumb. In some traits, you could even go lower, but 150 to 200 should be sufficient when you're working with a biparental mapping population. The experimental design, really similar to what I mentioned in the association mapping approach, should be replicated. Take care of different seasons, environments, to ensure that you're really capturing what affects the environmental components that affect this trait. And again, the genotyping approach here, similar to what I mentioned before, you can use SNP chips, you can use targeted sequencing, but you can also use scheme sequencing to generate your data set. There are two kinds of mapping approaches. You can do single marker analysis, which is too simple. It's just a simple regression. You take one marker at a time and do a regression with the trait and see if they are co-segregating. It's too simplistic. And also, you'd never, ever end up knowing the size of your QTL because you don't know the arrangement of the markers and all that. So it's too simple. And in most cases, nobody uses single marker analysis anymore. But the upside to it is you can actually just use Excel to be able to analyze that kind of data set. It doesn't require sophisticated software to run. The most recommended is multiple marker analysis because it allows you to use more than one marker at the same time and do a regression of those markers with the trait. And so it has more statistical power, and you can end up estimating the size of the QTL because you know where between which markers your QTL is likely to be located. Now I'm giving here an example of two software that we've used in the past a lot for biparental mapping. On the top, using ICIM for two particular traits, and this is the kind of output you get. And it's pretty much similar in QTL cartographer. Now, in this biparental mapping, the LOD score is usually the cutoff, and the cutoff that we tend to use is 2.5, between 2.5 and 3. And so anything above 3 is pretty good confidence that you have your QTL there. But again, we combine this with phenotypic variation explained. And in most cases where we have good QTL score and we have more than 10% phenotypic variation explained, that's a marker that we want to look into a little bit further and perhaps develop markers for indirect selection. So you've used these two populations, the diverse set or the biparental, you've identified QTLs. What next? Because you want to go all the way to the point where you're using these markers or these QTLs to improve varieties that you end up releasing to the farmers. Now, after you identify the QTLs now, you need to potentially identify the candidate genes in that region and maybe end up validating SNPs that you can now use either for integration or just selection for the trait in a wider population. Now, the way that you identify candidate genes is if there's whole genome sequence available. And for most crops now, even for the dryland crops, we have reference genomes available. If there's no reference genome available and you have maybe sequences of that particular region, you could do searches of public databases to be able to figure out, what sort of genes might be present in that location. And that seems to work really well because now there's a lot of data set in public databases that you could use to confirm the genes of interest that you have. If you don't have sequences for that region, it's also fairly simple to generate sequences, because when you're doing your association analysis, you'll have identified different markers, and those markers, you probably retrieved them from some sequencing information. You can use that to stitch the pieces together and design primers that would amplify that location in your target species, and use that to search public databases for what typically would be the gene in that location. And I say it as though it's so simple, because sometimes you can have a really big QTL of maybe even more than 100 MBs. In that case, you need to stitch several pieces together. And that's why, one of the prerequisites I talked about initially was that having a reference genome is a plus when you're doing trait discovery, because if you don't have a reference genome, it becomes a whole nightmare later on to be able to identify, for example, candidate genes, if you're interested in identifying candidate genes. But remember, you can still end up using these markers for selection, whether it's genomic selection or marker-assisted selection, without necessarily having to know what are the candidate genes in that region. So let's say you have a reference genome. How do you go about identifying potential candidate genes? Well, the typical thing that we do is, once we identify markers that are associated with the trait, we also do an LD analysis, a linkage disequilibrium analysis for that set of markers that we've used. And that allows us to know how many groups of markers are co-segregating. That tells you that if you find marker X to be associated with trait Y, it's possible that the other markers that are in linkage disequilibrium with this marker might also be co-segregating with that particular trait. And that also means that all the candidate genes in that region, that LD block, is also likely to co-segregate with that trait of interest. So the way that we identify candidate genes, once we identify markers that are associated with the trait, is basically go and look at all the candidate genes that are present in the LD region of that marker. And yeah, I was just giving an example down here, some of the candidate genes. So for example, in this particular case, you've identified an interesting marker here. It's very consistent across for this trait. The p-value is really, really good. And now you can go into that particular locus based on the LD that you've calculated and identify candidate genes, both upstream and downstream of that particular marker. Now, if you identify candidate genes and you find something that is so obvious, so for example, you were looking at, I don't know, oil content in groundnut, and you've identified this locus, and you go there and you find obvious genes that are involved in oleic acid production, and there are publications confirming that that gene is actually involved in oleic acid analysis production pathway, you can right away go into further characterization of that candidate gene just to see a lot more of what you can do with it. And so allele mining is one of those things that now we do a lot more. When we have identified a candidate gene, there's overwhelming evidence that that candidate gene is involved in the trait you're looking at. You can go into allele mining. And some of the other things that I didn't really find time to be able to discuss in this lecture are things like looking at haplotype analysis, which is immensely useful. I decided to just focus on allele mining for this particular case. And so what you do then, you go into that particular gene, and then look at, so you would have phenotyped a population where you know, okay, these are the individuals that are having, if you're talking about high oleic content, for example, you know, this is the group of genotypes in this population that are having high oleic content, and this is the group that have low oleic content. And so how you end up doing allele mining is then amplify regions of this particular gene and see if there are different allelic variations within the gene that you can actually associate with the trait of interest. And this is also now quite easy to do because you can generate the sequences really, really easily. And so especially if you have a crop for which the reference genome is available, again, you can generate, you can design primers, amplify, or even use targeted sequencing, amplify the region and generate the phenotypic data sets you need and do allele mining on that particular gene of interest. Now, remember, allele mining doesn't have to be on just one particular gene. You may find quite a number of genes in that same locus. It might be a cluster of genes, or it might be a group of genes. So you might end up doing allele mining, not just for one gene, but for a class of genes or for a group of genes. And it's highly useful in defining or narrowing down your association to a particular region and a particular gene that you eventually can recommend either for gene editing if you have resources to do that, or functional characterization, because then if you have overwhelming evidence that there's a specific mutation in that gene that is causal to the trait you're looking at, you're good to go in terms of doing functional characterization for it. Yeah, so I mean, in this case, I'm just giving an example of where, let's say you've already identified, you've done allele mining and you've identified a particular mutation that is associated with a trait of interest. Now, the example I'm giving here on the screen is for a gene that is responsible for disease resistance in sorghum. Now, in this study that was published in 2022, they actually looked at, if you see this slide, if you see the top sequence is for susceptible lines, they seem to have this deletion, and the resistant lines that have that insertion there. And here as well, you see that in this particular region as well, the susceptible lines have these particular alleles that are different for the resistant lines. Now, you've already done that allele mining. You can even go further, look at the haplotypes and all that. But in this particular case, these four, or the two, 1, 2, 3, these four loci, you can go ahead and design primers for them that you can straight away use for your marker-assisted selection, or you can introduce them into your genomic selection panel and use them for prediction of phenotypes for resistance to this particular disease. Now, I know I'm running out of time, as I had promised that we were going to have a little bit more time for questions and answers. I'm going to try and go through the next slides really, really fast. And one of the ways that you can use those markers is, to design allele-specific primers that you can use, or you can put into a low-density platform that you can use for selecting for the trait of interest. And this is just to show how, allele-specific primers are designed. This is not something that you really have to master, because there are several service providers out there that can do it for you. So as long as you've identified your loci, so for example, in this case, you know that there's a SNP in this region, a SNP in this region, and a SNP in this region, you can then go ahead and design allele-specific primers. Now, in this particular example, we also have insertion deletion. Now, allele-specific primers, for that reason, you really can't use indels for them, so they're mostly for SNPs. So in this particular example, you could only design this particular locus, this one, and perhaps this one as well, but not where you have the deletion, because they don't work really well with indels. So yeah, I mean, just to show, you would design CAS markers and then use them for regular phenotyping, genotyping of your set that you use for selection for your trait of interest. And this is just an example of, a data set that we generated of the kind of results that you may expect to see. So for example, in this particular case, you see if these were susceptible genotypes down here, you can actually see the difference. They have different alleles, and just by looking at them, you can select the ones that have the loci that you need. And in some cases, you can select for all the 3 or 4 loci that you need at the same time. If you want to have the full dosage, so you're looking for either progenies that have all the loci, then you go and select the ones that have all the loci. In some cases, you only want at least 2 of the loci. Again, you can go back and select for the ones that have at least the 2 of the loci. Again, you can also use such markers for confirming hybridity testing. In this example, I was also giving, now you've crossed the 2 parents, parent 1 has this differences in alleles. Again, you look at the progenies and use it to eliminate the ones that were not successful crosses. And you can do that pretty easily with allele-specific primers. Now, I'm going to summarize and say there are exciting opportunities to work with dryland crops in terms of developing resources, because these are crops that were not, the resources were not developed previously. There are several traits that remain uncharacterized, and especially the climate resilience trait, we are barely scratching the surface. So there's great opportunity to look into them, characterize them, and discover a whole world of things that we don't know and don't even exist in some of the major crops. As I did mention, there's need for a careful balance between phenotyping and genotyping, and I stress, with all the costs of genotyping going down, phenotyping is going to be our biggest challenge of this century. So we need to come up with really good, accurate phenotyping methods for climate resilient traits. We do need collaboration across institutions. One person cannot do it alone. I mean, if we're going to need more replications, more environments, then the more institutions involved, the better for us. And again, we do need to build capacity so that we have more people who are understanding what we need, the biology, those who are doing physiology, those who are doing genomics, those who are doing agronomy. We need everything working together. And we do need to also include functional characterization for some of these traits so that we can actually fast track the process of breeding and introgressing traits of interest into farmer preferred varieties, which is ultimately our objective, our major objective. Beyond the academics that we are understanding, we need to give something to the farmers. And I also just want to mention that in my group, I have opportunity to host postgraduate students. If anyone is interested to come and spend a little bit of time understanding these interesting crops and the wonderful research that we do, I'm open to collaboration. And thank you so much for listening. I hope we still have time for questions and answers. Thank you so much.
Thank you so much, ma'am, for this wonderful lecture. So I'm requesting all the participants, if they have any query, they can ask one-to-one. I'm like giving permission to all, so they can unmute themselves.
बोलो प्लीज बोलो पैर छू रहा हूं
In case someone is talking, I cannot hear.
I request everyone who would be having any doubt in this session, kindly mention your question in the chat box so we can ask the question to the ma'am. Till now we have got the things like your session is very, your PPT is very well, ma'am. Your presentation is very well.
So I see a question. In case of F2
Rohit sir, are you there?
generation,
Yes, ma'am.
can you hear me?
Yes, ma'am. Yes, ma'am. Yeah.
Okay. Should I respond to the question on
Yeah,
on the
ma'am, we are
F2
able to hear you.
generation,
Yeah,
or
ma'am.
should
We are
I?
getting questions in the chat box, so you can like open chat box. You can like solve the queries, I think. Or
Okay,
even
so there's a question there. In case of F2 generation, there's a drawback. Both traits, dominant and recessive, come in 50-50 possibility. If farmers sown seed which are come from F1 generation, is there any effect on their productivity? So the examples I was giving were very specific to trait discovery process. It's for you to discover what is associated with the trait, then you introduce it, and then end up releasing the variety to the farmer. I do not expect you to release an F2 to the farmer because obviously this is still not fixed. When you do varietal release, we can have another complete lecture on this. When you're doing varietal release process, it's a completely different phenomenon. It has to be fixed. It has to be tested that it's stable before you release it. That's how I would respond to that question. I see someone else has their hand up.
Ma'am, there is one more question from Ankush sir. That is, can we use this technique to develop drought and salt stress resistant in forest trees also?
Yes, absolutely. So if you saw the examples that I gave, you can use genome-wide association studies to look at association among trees. And all you need to do is sample a diverse set of those trees, look at how they respond to salinity, and you can use that data set to generate also genotypic data set and look at association of different markers to salinity tolerance, for example, if you've phenotyped for it. Now, because trees are not something that you can replicate across different environments, you can take advantage of different seasons. So you can phenotype across different seasons to look at how they are responding to the salinity that is imposed on them, and then use that to do association mapping. I hope that in the particular case that you are referring to, that you actually can have enough trees that are varying for the trait for a response to salinity that is sufficient for you to map that trait with confidence.
Ma'am, we have one more
Does that answer your question?
from Dina.
Yes.
And yeah, that is, what about the pest resistant crop like pigeon pea? Are there any variety which is resistant to pod borer complex of pigeon pea?
I know that has been quite a big discussion. I think among the cultivated, there are very few, or maybe there isn't any that is resistant to the pod borer complex. But I do know that now there's an effort to look into the wild relatives, and there's promising data from wild relatives that they actually have some level of resistance to pests. And so that is being introgressed into cultivated germplasm. And in some cases, it shows promising results. But ultimately, now that we do know that we can use genetic engineering, that's an option that you can look at. Also to mention that even though we expect that we're going to find some level of variation for traits of interest among the wild relatives or distantly related germplasm, it doesn't, you don't lose anything by integrating different approaches to enhance the trait. So if it means bringing in resistance from the wild and also doing genetic engineering, doing gene editing, if there's reason to do that, doing mutation breeding, all those together to come up with a solution, it can only enhance your chances. So why not go for it?
I think you have well explained that thing to our participant, ma'am. And more of the participants are excited to know your email ID and your PPT, as they find it very informative. More are interested to have your email so they can request you for the postgraduate studies and they can get your guidance. As you
डॉक्टर
have
ज्योति
already
आपके
guided
साइड से
more than
नेटवर्क वीक
250
है, आप वीडियो
students,
ऑफ कर लीजिएगा,
more many
आप ऑडियो
students,
में
at
बात
least
करिए.
50 students you have guided, more from different nation under your headship. So they are more excited to have you that thing. We are request you on the part of I just request the ma'am to just share the email ID of yours so that our participant can request you for the postgraduate study. For that, I'm very thankful on the part of AgriMeet Foundation. And
yeah, your line was breaking quite a bit, but I think I understood the request. I'm typing there my email address so you can share with everyone.
Yeah.
And yeah, feel free to send me a message or to send me a LinkedIn. I'm also available in LinkedIn. I'm more than happy to follow up on any specifics with anybody.
Yeah, ma'am, we would be very thankful if we get that email ID from yours and get it for a participant. Further, we thank you on the part of AgriMeet for providing.
क्वेश्चन चैट बॉक्स में देखिए कि कोई रिमेनिंग क्वेश्चन तो नहीं है.
Okay, sir. So there is nothing like a more remaining question, but ma'am, I have shared the her email ID. I think two participants are very excited to have the ma'am ID. I just request all the participants to send only the valid questions to have theory from ma'am and valid proposals for postgraduate study. Further, thank you, ma'am, for today's session. I further welcome and thanks Dr. Sunita, ma'am, senior scientist from ICRISAT, to be there with us. Thank you, ma'am, for joining us for the today's session. And thank you, Odani, ma'am, for today's session. Thank you so much.
And thank you also for the opportunity to give this lecture. Thank you for considering me, and thank you everyone who was listening to me. Shout out to Sunita, nice to see you here. Thank you all.
Ma'am, one more request. Sorry, at the end of the session, we are saying to you. Yeah, here we have got the request from many participants that if you have the time, they all are ready to have one hands-on training or training session online with you on genome editing, because they are very excited by the way you explain everything regarding climate resilient crop, how they can develop, about gene editing, about allele mapping, everything. They are excited. In case you would be available, we would be happy to have one training for all the participants throughout the world like these in the future. Thank you, ma'am.
No problem at all. Let's plan it. I'll be happy to do some that training, and I'll also be happy to bring in a few more experts to add their value as well. So yeah, happy to do that.
Okay, ma'am. Thank you so much, ma'am, for today's session. Thank you.
Thank you so much,
Thank you
Odani
all.
ma'am, and thank you so much, Dr. Sunita ma'am.
Thank you all. Bye.
Bye bye.
Bye.